Conservation Sciences Fall Seminar Series for 2021. Today we have Dr. Daniel Pauly, and Dr. Daniel Pauly is a world-renowned fisheries scientist and serves as one of the board of directors of Oceana. He is currently the principal investigator of the Sea Around Us initiative, a large research project devoted to identifying and quantifying global fisheries trends. He is also a Killian Professor at the University of British Columbia's Institute for the Oceans and Fisheries. Here today we have Dr. Daniel Pauly talking. And if you have any questions, if you throw them in the chat box, I'll read them off to Dr. Pauly, but if you could unmute yourself at the end, that would be awesome. And Dr. Pauly, I'll turn it over to you.
Yep. So I will share my screen and here we go. So I will talk about something that everybody will agree on, that fish must breathe. But actually, people don't take this seriously, that fish must breathe. And there is no real theory that links the various aspects of this breathing together. And I have attempted to come up with something. I call it GOLD because it sounds good. And I will take you through this. The point is that a liter of water, or a gallon of water if you prefer, contains 30 times less oxygen than air. This is surprising, but this is the case. Then the water is far more viscous and dense than air. So you have to spend lots of energy moving it around. For example, if you are a fish sitting like a pike and having to bring water across your gills. And then diffusion of molecules is immensely slower in water than in air. I don't know why I have in air than in air. They're in water than in air. So gills, fish have over millions of years developed super duper gills to overcome this problem. And if we look at the thing that maybe a fish that lived hundreds of millions of years ago, it was little, and it had apparently, given the reconstruction, very inefficient gills. At least as compared with the present situation where carp, that's the fish that you don't like in the US, but there it is, the head is full of gills, or basking shark, that also the head is full of gills. So you realize when you see these fish that breathing matters to them. Well, this is actually not the case in Arapaima. This is the biggest freshwater fish, and it has a very small head. But this is an obligatory air breather. It has to come to the surface to breathe, and if it's prevented from doing so, it will die. So this is not a fish in a sense that I'm talking about here as water breathers. And I know that this is not a fish either. This is a French car, a classic French car. You can see it in gangster movies a lot, in old gangster movies. And it has in the front a radiator that is supposed to help cool the engine. And how does it do that? Incidentally, this is not age that makes me talk about reservoir, radiators. This is pertinent to the talk. So you have hot water coming from the engine, and it needs to be cooled, and it goes through the radiator, through lamellae, which expose the water through a thin metal membrane to air. And this air comes from the front, and it picks up the heat, and it then it cools the water, which then goes back to the engine. Now imagine you need to have a bigger engine. this is a gangster movie. You have to escape the police. You have to have a big engine. How do you do that? Well, you can make it wider, you can make it higher, but you cannot make it deeper. You cannot make it deeper because the air that comes out of the radiator is already hot, so it cannot work if you do a radiator behind a radiator. So basically, there are 2 dimensions available for cooling. Now look at gills. There is they are lamellae, like in a radiator, and the water that comes in is loaded with oxygen, and the oxygen is picked up, and the water that goes out of the gills, or that across the gills, is free of oxygen. Not completely, but functionally, this is not worth picking at. So basically, there is no point putting gills behind gills. There are only 2 dimensions available for the growth of gills. That means that the volume of a body that has to be supplied with oxygen will always grow faster than the surface of the volume of the fish. And this is I did one time a paper for a science journal for kids that I do recommend, and basically it explains that using a very small cube fish that has a large volume surface to volume ratio at the bottom of the table, and the same cube fish has the a lower surface to volume ratio. Now that is what can be explained to kids, and then a parent should understand that as well. So because of this, and this is inescapable, fish, when they get bigger, have less oxygen, get less gill area per unit weight, and thus less oxygen supply per unit weight. Because the oxygen that goes through the gill is proportional to the surface area. That's fixed law. Therefore, we can always conceive of a weight, I call it weight infinity, that is so high that the metabolic rate, that is the oxygen that is supplied, is just sufficient to maintain life. Never mind how we define life, whether that implies them running around or them doing things and stuff. The point is that whatever they want to maintain can be maintained at this level, but not beyond. You can, yeah. So, is this empirically verified? Yes, it is. The Japanese researchers have again looked at the surface area of carp, of gill carp, and noted how fast they grow. And in larvae, they grow very fast. The surface area of the gills and the surface area of the body combined lead to a growth that is faster than the volume. So the slope is 7. So the small fish are not limited by oxygen at all, and their growth therefore is exponential. I will not discuss that here, but the growth of fish larvae is exponential unless they die. Then obviously good things have to end, and the gill just keep up with the volume, one. But the point is that as adulthood or the juveniles are reached, and then this always drops to less than 1. And because a surface cannot keep up with the volume, and that is 0.8 in carp. And you can see that's empirical, the points that you get out of this. If you plot them against body weight, you get a decline, and the fish at some point has to stop growing because it just gets enough oxygen to grow. So what happens given this in A? What happens if you increase the temperature? Well, fish being ectotherms will have a higher body temperature, and for reasons that I will explain in a minute, their metabolism will go up. The oxygen requirement will go up. And therefore, given that there is no change in the anatomy, in the gill anatomy, they will have to remain smaller. So increase the temperature, the fish will have to be smaller simply because the oxygen that is supplied to them is not is just sufficient to reach a smaller size. And this is well known. This was noted by Putter in 1920, and it can be shown, for example, in cod. And I could take any fish that lives north in Canada, compared with a fish that lives in the southern United States, and you get a curve like this for the growth curve, with the cold one being bigger but growing slower, and the warm one growing fast toward a smaller size. So, and this is also well studied well known to the people who study the taxonomists, that fish live longer and grow larger in the cooler part of their range. I don't need to elaborate on that. You will know it yourself. And we have shown that if you extrapolate this into the future, you get a massive reduction of size throughout the world in marine water, but it's also true for fresh water. So yeah, and now the fun, this simple stuff is over, and I will begin now a more boring part, because I have to explain why this is like that. So basically, growth rate is a function of two processes, and I don't know what happened here. One that is positive and one that is negative. And the positive process is synthesis. Synthesis of body protein requires energy and building material. The building material are amino acids that are synthesized under ATP is used for that, adenosine triphosphate, and for this oxygen is needed. So the power that links body weight and surface area, I call it D for Daniel, that's easy to remember, is always smaller than 1. So it's weight to a power smaller than 1, for example, 0.8. So that's protein synthesis. The next, and this power smaller than 1 can be about 2/3 in small fish. That's based on the experiment that von Bertalanffy did. Von Bertalanffy was the fellow who proposed this growth curve, and he proposed this growth curve based on experiment that he did with guppy. And guppy really have 2/3. There is fish that are very small that have even a lower ratio, a lower D value, and most fish are about 0.8, 0.7, and the highest is 0.9, but it's always smaller than 1 because it's a surface. And the negative process involved in growth is protein degradation. But this protein degradation has to be understood. It is not the burning of protein, because this burning requires oxygen. It's simply the loss of the quaternary structure. What is the quaternary structure? The molecules, as you well know, have a primary structure that is the sequence of chemicals, and then they have a secondary structure. They are helix, and then the helix bind, and then twist, and this twist themselves twist, and that's called a quaternary structure, and it's held together by very weak bonds, which can be easily broken. And as soon as a molecule loses its shape, it cannot be used anymore. Even though it's already, the chain is still the same, it doesn't have the right shape. So it has to be resynthesized. And this process of denaturation, of loss of the structure, is driven mainly by Brownian motion. And that's the reason why the temperature influences it. And molecules have in the body, molecules in the body of an animal last from a few hours for certain molecules to several weeks, but they have half-lives, and after a while they must be renewed. All of them, they must be renewed, and the renewal of them is the reason why even a non-growing fish must synthesize just to keep its pool of native protein going. So once a protein is denatured, that is, it loses its quaternary structure, it becomes part of the amino acid pool to which the food contributes, and it is the fuel for the energy metabolism. No problem there. And you can see that the energy metabolism can be only a certain quantity because it needs oxygen and amino acids. So basically, protein synthesis competes for oxygen with various activities. So breathing, swimming, foraging, digesting work. For example, if food is scarce, at the end of the day, the fish might have the same quantity of oxygen, of food in its stomach, but it will have spent more energy looking for it, and therefore protein synthesis would suffer. Okay. So this model that I have shown you explains quite a few things that I will now present to you. For example, it explains why growth is asymptotic, because growth becomes more and more difficult to realize as time goes, but the pressure that I call it dimensional tension between the gill's surface supplying oxygen and the volume requiring oxygen, this exists from the very beginning, from a juvenile on, and it becomes worse until the point at which growth cannot occur anymore. Now, it also explains why in over 70% of the fish species that exist, and you can assess that by looking at FishBase, which you may know, that has a database of all fish in the world. About 70 to 80% of the fish species, the female get bigger. The female get bigger than the males, though, and yet people tell us that fish stop growing because they invest in reproduction. But if this were so, the female, which do far more for reproduction than males, would be smaller, but they are bigger. So that contradiction can be explained easily by the fact that females are a tiny bit more quiet than male. When you breed for fast growth, you breed for quiet fish. And basically because of this competition between synthesis and activities, if you shift it a tiny bit toward synthesis, a bit less activities, it will have a huge effect, because about only 5 to 10% of the oxygen and actually the energy absorbed by, taken in by fish is used for synthesis. The rest is activities. So if you reduce activities by a tiny bit, it has a huge effect on growth. So this also explains why the food conversion efficiency of fish declines. If you read the literature, King and others, they try to explain, they give lots of explanation for why the food conversion efficiency of fish decline with size. And it's simply that they have no oxygen when they get big to convert the food into growth. And so you can use different equations to express this, and basically it tends towards zero, the food conversion efficiency, which at the maximum size, a fish eats but doesn't grow. Then the food conversion efficiency is zero. It also explains, this thing also explains why there are daily rings in larvae. In fish larvae, you will know that you can tell rings that are daily. I'm not talking about the annual rings that allows you to tell the age of fish in terms of years. I'm talking about rings that are laid daily especially by larvae of fish. And you can count this thing on a very good microscope. And the reason for that is, for the laying of rings had never been properly explained. And I explain it as by a fluctuation of pH in the body of the fish, and specifically in the lymph surrounding the otoliths. Basically, when a fish is active, it runs out of oxygen and the pH goes down, the pH of its body lymph. And then when the pH is low, the substance that is deposited in the rings, in the otolith, has a different appearance than when oxygen is high. So basically, I think the more when oxygen is high and pH is low, high, the more organic material is laid down. And so you have a ring that is produced daily. On the other hand, when the fish gets big, basically it is under oxygen stress all the time, and you cannot see rings anymore. And that's the reason why when you look at older fish, you cannot see their rings anymore, but you can see them in larvae. This also explains why fish seek colder, deeper water when they get bigger, because that's the only way they can continue growing. They go into deeper water. And so when you study in Germany, they tell you that it is Henke's law. This is one of the guys who has described this first with place in the North Sea. The North Sea, you see the UK on the left side, and Holland and so on, and Denmark on the right side. And you see this little place that are in shallow hot water, and the large place are in deeper water. On the right side, you see two fish from Hawaii that get larger as they grow, and as they get into deeper water as they grow, and the deeper water is colder, and that allows them to continue living. This is also the reason why when people growing, why people growing fish in aquaria, not in aquaria, in aquaculture, have to consider aerating the air, aerating the water of this animal they have. And I have here photos from China, intensive culture of fish in ponds, high tech, and you see that an incredible energy investment is made, costly in terms of money, to bring oxygen into the water. Now, why do they do that? Not only to prevent the fish from dying when the oxygen level goes down early in the morning, but also because you have seen the food conversion efficiency depend on the availability of the oxygen. The availability of oxygen is low in a big fish, but it's also low when the water contains less oxygen. So there is huge amount of money that is spent on aerating, because otherwise you don't recover your investment in providing food. Here you see fish migration. In A, you see fish from West Africa. In the south, you see a country called Senegal and the Gambia. In the north, Morocco, and in the middle, Mauritania. So they go from Senegal to Morocco and back. Why the hell do they do that? Well, they try to stay in the same temperature because they cannot afford to stay at the same place. They are too big. The small one can stay at the same place because they can handle the hot temperature, but the big ones cannot. And so they migrate back and forth to prevent themselves from being cooked. Now, this is also the reason why fish are predicted to move, and fisheries are predicted to move poleward. And this map actually is the first that the IPCC published, showing this phenomenon on the ocean. This is well known for plants. This was well known for terrestrial animal, and we did this for fish and fisheries. Basically, and you know this process. It's happening that you get in New York fish from Florida. We get in British Columbia fish from from Baja California, and you get in the Midwest fish from the southern states. That is happening on a grand scale, and the problem with this is that there is a tropical band of fisheries that will go down the tube because the fish are leaving. However, this is a prediction, right? This is a prediction in 2050. But global warming has begun several decades ago. You can see that the increase in temperature that was reported by the Hadley Center from the UK. And so we invented a measure for the movement of fish that we call the mean temperature of the catch. And the mean temperature of the catch is basically based on each fish having a preferred temperature. That is well known. And what we take is this preferred temperature is kind of conservative. They don't change the preferred temperature in the context of years and decades. This is more hundreds of years and thousands of years. So you can compute for any catch, the catch that you see here, or the catch of a country, or the catch of a state. You can compute the mean temperature of the catch by taking the catch of one species and its temperature, the catch of this, and then you compute the mean temperature of the catch. You can see that, and the mean temperature of the catch, in tropical in temperate system, You can see that in the upper plot. In temperate system and subtropical system, and I'm talking about globally now, the mean temperature of the catch actually is more or less parallel to the mean temperature of the environment. And here with UC surface temperature of the sea. And in the tropics, though, that is not so. In the mean temperature, the actual temperature, it increases. But the mean temperature of the catch will increase at first because a few fish leave the tropics that have colder water affinities. But after a while, the mean temperature of the catch cannot increase anymore. And that's the reason why the fisheries go down a tube, because in the tropics, because there is no fish that are replacing the one that are coming from, that are leaving, because it's too hot. And because you guys are in freshwater, I have added to a standard lecture a paper that I just did with a bunch of Estonian colleagues. Now, for those of you who have problem with geography, you see that little map. The big thing at the upper left corner, that is Scandinavia, right? This is Sweden and Norway and Finland, and the little square in the south of it is the Estonian Russian border. And this lake is very well known from a film called Alexander Nevsky. You should look at it. It's superb. It's an anti-Nazi film that was done in the late 30s. And anyway, in this lake, the temperature was monitored, mean temperature of the month, mean monthly temperature, and you cannot detect a real signal. But the fish, the fish community, it changes radically. And in the late 80s, all of a sudden it began increasing. That is the model here uses a simple segmented regression. And while until the 80s, the changes were without direction. The changes, the mean temperature of the catch. But from the 80s on, it increased rapidly. And this is the first application of this thing, of the mean temperature of the catch to fresh water. And I presume that this is happening in several of your lakes. It is happening in all over the place. In China, for example, I have done with Chinese colleagues, the South China Sea, the East China Sea, and the Yellow Sea in the north. And the Yellow Sea had the strongest effect. The South China Sea had no effect. The mean temperature of the catch remained the same. And so that is the prediction are really validated. So basically from that, you can infer the following. In subtropical ocean and temperate ocean, waters, I should say, also it's also true for freshwater. On the left, you see you have fish with cold water affinities, blue, and they tend to not do well. And for example, salmonid, and they are replaced by fish with warm water affinities. And in the future, that effect will be much stronger. In the tropics, though, and that's true for freshwater also, you start with tropical fish and a few subtropical fish. The subtropical fish don't like it and disappear. And at the end, you have only a few fish that are really, really temperature tolerant. And but you have fewer of them. And that seems to be what is happening, and on a grand scale. Now, let's get back to the figure, and that is the last point I will explain, and that is not easy, so try to engage. Basically, if this is true, that the relative oxygen supply declines with body weight to the point that the fish cannot grow, they have to spawn at a smaller size. That's, we know, is the case. And I call Q the metabolism at the metabolic rate, or relative metabolic rate, and QM is the metabolic rate at spawning and at first spawning, at reaching first maturity. And Q infinity or Q maintenance is the metabolic rate they will have when this is over. It's equivalent to the at rest, to the metabolic rate at rest. So you have a ratio between these two. You can see it. And if you increase the temperature on them, the W infinity will be smaller. We have agreed on that before, and it can be shown empirically to be the case. This will imply that QM is also smaller. So the question is the ratio between QM and Q infinity the same in both graphs? And you know what? It is. In 84, and I wrote a paper that showed small gobies, small intermediate fish, and big tuna having a ratio of 1.36. And I was very proud of this, and nobody gave a hoot about it because it's weird. And so I decided to look at it again after several years, and I got the same thing for tilapia. And more importantly, I got the same thing. Colleagues working in Idaho got exactly the same thing. And that brings me back to this. Now, this, when you look at, if you ask the question, where do fish reach maturity when they do? If you're a professor, you look at the textbook, or if you're a student, you look also at the textbook. If your question is, why do fish reach maturity when they do? The answer will be something like this. The environmental stimuli, they are environmental stimuli, and they affect the hypothalamus, it goes into the pituitary, and then it goes into a cascade of hormones. And this is a picture from 94 in A, and B, a picture from very recent 2017 or something, a major textbook. So that's supposed to be the explanation. And it is the explanation when do fish reach maturity. However, if you look at fish that get very old, they spend a huge amount of time not perceiving the environmental stimuli that the adults do. For example, orange roughy, 34 years. They reach 44 years before they spawn. White sturgeon, 11 to 34 years. The Amur estuary Kaluga is also a sturgeon, 21 years. The lake sturgeon, 20 years. What prevents them from starting to spawn earlier? Because they do see the same thing that the spawning ones see, but they don't spawn. Another way of presenting that is here. The cascade, the environmental cascade, is shown. It occurs every year, right? Sorry, not the environmental cascade. The stimuli occur every year, but they are not seen. They are not seen by the young fish, even though they have eyes and they have a pituitary and a hippocampus and everything that is being mentioned in there in this little drawing. but they don't see it. And then something changes. What is it? Well, you have to look at the way we perceive the relationship between growth and maturity. The conventional view in A is that fish grow fast as juvenile, and then they mature, and then they don't grow anymore because the energy goes into reproduction. That's what we learn, and that's you can find it in hundreds of papers and textbooks. Actually, this is the fact that we use length here, growth, mislead us, because if we were plotting growth in weight, we would see that the spawning occurs before the growth is maximal. Actually, in most fish that are about more than 1 kilo, that for small fish, something else happens, but for larger fish, most of the fish that you work with, sport fish, the spawning in weight happens before the inflection of the growth curve. That means they spawn and growth accelerate. And the inflection rate, the inflection point of the growth curve is occurring after spawning. So it cannot be that growth is reduced by spawning. I implore you to plot a few growth curve for large fish, and you will pike, perch, and so on, and you will see that the growth curve, not like in A as length, but in B as weight, and you will see that the inflection point of the growth curve, where the growth curve is maximal, where growth is most rapid, is actually after they have spawned. So the spawning cannot be the reason why they stop growing, because they actually don't stop growing after they have spawned. And rather, what happens is when fish get bigger, the oxygen supply per unit weight drops. You have seen that earlier for carp. This is simply because of the dimensional tension. And when it is a certain fraction of the maximum of the lowest metabolic rate that they can have, which is at maximum size, they start spawning, and that is about 1.35 of the maintenance metabolic rate. And the maintenance metabolic rate is the metabolic rate that they have when they quiet, when they do nothing. And they can therefore monitor their own metabolic rate and can tell when it starts to drop to 1.35, that metabolic rate, it's time to spawn. Because if I wait longer, I will not have free oxygen to produce gonads. And in A, you can see my original plot, which is now corrected for phylogeny, and it's not 1.35 anymore, it's 1.32. Here you can find the work of this colleague in Idaho there, and they got 1.35. Here you got fish in Turkey, freshwater and marine fish. Here you get, indeed, you get fish in China, and this is more than 100, but we could, 96 where we could deal with the phylogeny, and with the 1.41, and the tilapia that I just showed you, 1.35. So basically, and there are more under review, and also it seems to apply to invertebrates. Basically, then, and I'm almost done. Basically, the standard view of what triggers spawning, which is environmental stimuli, influence the hypothalamus, influencing the pituitary, and so on, is a sufficient condition, but a necessary condition for spawning. And what triggers it off, or what opens the faucet here, is that the fish must have a certain size, and that size relative to the size they will reach is the trigger. And if you don't have this trigger, you don't have a start of this. And the mechanism that we are taught is not self-starting. You need something to start it that is outside of the hormonal setup. And so the fish has to monitor itself to know it is now time to look at environmental stimuli. Before that, there is no reason why the environmental stimuli should be having any influence any more than our children are interested in sex before their internal growth has reached a certain level. So basically, you have a necessary condition for something to happen, and then what we have been learning is not only the sufficient condition. So my end, the end of this is that what I told you, even though it is, I'm sure it is strange, are not just those stories. I have been, every step on the way is documented in the primary literature, and you can get the paper, PDF of the paper, simply by writing me. Thank you very much.
Thank you, Dr. Pauly. I have a quick question. So how does this hold up for like endothermic fish? So I don't know, maybe like tunas or something like that.
Basically, it pushes the performance level a notch higher, but it doesn't change anything. The endothermic fish also have the same constraint. And in the paper that I distribute to explain this, tuna are among the various fish that it doesn't work. It doesn't work as soon as you start breathing air, because air implies other constraints. For example, the fish, the arapaima, the huge freshwater fish from South America, it is limited by what kills it at the end is the fact that it would have to get up all the time to breathe and get down and get up and get down. So it would have no time to do anything else. That ends up being limiting for it. But this applies strictly to air breathers or air breathers that don't bother with gravity, because as soon as you are outside of water, you have different constraints, and you are also affected by dimensional tension. But the dimensional tension is of a different type. For example, you produce heat because you're a mammal, and you lose heat to your surface area. So you have heard of Bergman's rule, that the animals in the Arctic have to be big and reduce the surface area. So the ear, the snout, everything is shorter because they have to reduce the surface area to not lose heat. On the other hand, in the tropics, you have elephants that have a problem with getting rid of heat that they produce internally. And what do? They have big ears, right? And whales, they breathe air. They have no problem with getting oxygen out of the atmosphere, but they have problems getting rid of heat. So the Boston whalers, the people who killed whale before, they just run them to death, and they have heat. They produce heat internally, and they cannot get rid of it fast enough. They get rid of heat through their tail, through their fluke. So basically, the surface area problem, surface volume problem occur throughout the animal kingdom, but for different taxa, it manifests itself differently, right? It is the same problem, it's a geometric problem, but it manifests itself differently. Other questions? Yeah. So the question is the thing that I mentioned, that calmness allow you to grow faster, and basically, fish. This has been evaluated for very few species, but basically, they only 5 to 10% of the food they get and the oxygen they get is devoted to growth, to somatic growth. 90% is devoted to moving around. So if a fish moving around, displaying aggression, doing things. So if a fish doesn't move much, it changed a little bit its behavior. A huge amount of the somatic growth can actually increase a lot. So basically, when we breed, when we genetically select fast-growing fish, we select calm fish, and you can see that if you compare the salmon, European Atlantic salmon that are used in farming, that are farmed, if you compare them with salmon that Pacific salmon that are wild and have to forage for themselves and to defend themselves against all kinds of predators, you can see that the Atlantic salmon is calm like a cow in the field, whereas the Pacific salmon is doing things all the time. And basically, domestication is calming down. That's what the horses that we have now are not the wild horses. Wild horses, you cannot jump on them. They are impossible. And so domestication is calming down so that energy, food, and oxygen for fish goes into growth and not into activities. Now, if a female fish under the same condition as male fish is a little bit more calm at a level that is actually barely detectable, it will grow much better. And that's basically the thing, that the female are a bit more quiet because they don't spend so much time displaying how good they are. You have learned from Darwin's natural sexual selection that males actually display because they are chosen by the females, and they have to display, and display costs energy, and so the female tend to be faster growing because they don't have to bother with so much display. And a risky analogy, a very pregnant woman will not run marathon, right? There are things that very pregnant women cannot do and do not do because they're a bit more calm. That is the whole thing. Yeah. If you were doing what metric would you suggest? Well, actually, if you were designing a broad-scale monitoring program across multiple, what I would do, and you can do it already now with the data you have, you can compute the mean temperature of the catch of the community that you have and that you had, and you will see that the mean temperature of the catch is already higher now than it was before. That I will bet my bottom dollar that it is already happening, because I have seen in the 80s papers that say that in the Midwest, the community have changed. The fish community have changed toward fish from that are more warm loving than fish that are cold, with cold affinities. So the metric that I propose is the mean temperature of the catch, and of the catch or the community. And you assign to each fish a temperature, and then you weigh the total community or the total catch by this temperature times the abundance, and you will find that it changes. And if you want, you can use the paper that I showed you from Estonia, that as a model. It was quickly done because the Estonian had already published the community changes. That's the reason why I wrote them. They describe the community as cold loving and middle loving and warm loving. But these are words. This is not science really. And I proposed to them that they transform the community description into a number, and this transformation was done, and it led to the graph graphic that I showed you, and you can do that.
Just want to chime in and say that's super interesting, and we totally could do that with long-term data from Midwestern lakes. So thank you.
The next question is the lowest solubility oxygen is salt water, and so basically, for the theory, as I presented to you, I don't see the difference between the physics of freshwater and saltwater being very different. I know that there is less oxygen in saltwater and stuff, but the difference that are caused by temperature difference are so crude, are so strong that they seem to overwhelm the difference between the two system type. So I don't really look at difference between salt and marine water. I see the same mechanism. But I think that well, you might be tempted to think that deoxygenation is stronger in fresh water, but actually in marine water, in marine system, it's increasing very rapidly. And fish like tuna have a much thinner layers where they can be, because there is at 500 to 1,000 meters, in most ocean, a layer of oxygen poor or oxygen free layer, and this layer is expanding now, and so the problem of deoxygenation is occurring in both freshwater and marine waters. Yeah.
I think we probably have time for one more question if anyone wants to ask Dr. Pauly that.
Mind you, I would be, if you have no specific question now, I'm always ready to answer question individually. Yeah, this lake Estonia lake is very interesting because the mean temperature of the water did not really increase, but what increased is the frequency of of extreme event. And basically, the fish respond to extreme event, not to average, because the average temperature has changed very little, but the frequency of extreme event has changed. So basically, I think that's what it is. The fish are telling us something that our thermometer that do not tell us, because the frequency and timing of extreme event is what affects them much more than the change in the mean. Thank you.
Yes, thank you, Dr. Pauly. We appreciate you coming and giving a talk to our seminar series. So next week we have a presentation at the same time, 12 to 1. So tune in for that. Oh, we do have one more quick question from Sam.
Yeah, I was curious if you ever
Do you
needed
change?
to
Yeah, change in fishing practice. Actually, if you had a situation where a lake is exploited for its pelagic fish, and then the pelagic fish are not fished at all, and now we're catching only the demersal fish, the radical change, this would not work. But they made us, in a Nature article and in an Estonian paper, they made us, the reviewers made us deal with at length with the fishing practice, and it had no effect. As soon as a lake or a water body is exploited throughout the food web, if you exploit essentially all there is, then the community will reflect the temperature changes. So it was something that the reviewers wanted, and we did it, and we never found any effect of that. But we can think of such effect. Yeah, yeah. So I'm gone, and you are gone, and nice to have seen you. Bye, bye.
Thank you, Dr. Pauly.